We'll cover chapters 10 and 11 as a combination chapter, mostly to cut down on some of the time that we take covering this topic in class. There's a lot of information in both of these chapters, so I strongly encourage you to go back and read through, especially the mechanics of the processes of transcription and translation. In these chapters, we are going to cover the processes of gene expression, taking the information from our DNA and turning it into a phenotype. Now, that phenotype could be a functional RNA molecule, or it could be a protein that is going to produce a particular phenotype. Either way, the products of our genes are going to affect how those genes are expressed in our cells. We've spent our last couple of lectures talking about DNA, and now we're going to switch over to RNA. RNA is arguably just as important as DNA, if not in some cases more important than DNA. And the reason is that RNA has a variety of functions in our cells. Now, not that DNA's function is minuscule. DNA's function is to store, maintain, and transmit genetic information. That's absolutely vital to the cell. But RNA molecules take on a variety of different functions depending on their shape, which is odd because their shape is actually more simple than DNA. So one of the reasons that RNA is important is that the leading hypothesis for origin of life is that RNA was originally the coding nucleic acid. So in our first cells, it's thought that the coding molecule that stored and transmitted the genetic information was actually RNA. Now, the reason that scientists think that it was RNA is that RNA actually can function as kind of DNA and as a protein at the same time. And we just learned that during DNA replication, DNA polymerase needs a lot of help. First, we have to be able to make our DNA polymerase. We have to do that somehow. But then also, we need a variety of proteins in order for DNA replication even to take place. But remember that we talked about that enzyme called primase. Now, primase was an RNA polymerase. And RNA polymerases are basically a one-man show. They can come in and just immediately start opening up the DNA molecule and replicating it. And so because RNA can do some of the things that DNA does, but also do some of the things that proteins do, it's thought that it was the sort of original molecule that became a self-replicating molecule through the process of chemical selection and then became a coding and replicating molecule. And then eventually DNA would have taken its place because DNA molecules are a bit more stable. And if you want long-term maintenance of your genetic information, you probably want it in DNA. And so as evolutionary history trudged on, proteins began to take on more of an enzyme function and a variety of functions in the cell. DNA became the storage material, and RNA took on a variety of different functions. We've already talked about the basic structure of a nucleotide and a basic structure of nucleic acids. DNA and RNA share a lot of characteristics. They're both linked from the 5' to 3' direction by phosphodiester bonds between the 5' phosphate and 3' hydroxyl group of two nucleotides. RNA generally forms a single-stranded molecule. However, it can fold back on itself based on the rules of complementary base pairing. So we will sometimes see double-stranded regions in an RNA molecule that are following the same base pair rules. In RNA, we find the nucleotide uracil in place of thymine. So what that means is that in RNA, adenine is going to bind with uracil instead of thymine. Now they're still going to form just two hydrogen bonds in between them. And when we are making DNA, making RNA from DNA, we're going to have to take this into account. Our other major difference is that we have this hydroxyl group on the ribose sugar, which is why we call this a ribonucleic acid, because sugars are supposed to have OHs on all of their carbons within their ring. Our phosphate group is the same. Our nitrogenous bases are either double ring purines or single ring pyrimidines, again, with the exception that we've got uracil in place of thymine. Uracil is a pyrimidine, which means it has a single nitrogenous ring. So we've got uracil in place of thymine. We've got ribose instead of deoxyribose. And generally, the overall structure of RNA is going to be single-stranded, but in many cases, it will be folded. Not all, but many cases it will be folded. There are several major types of RNA molecules. The first type that we are going to talk about is actually messenger RNA. Messenger RNA is going to take our information from our DNA to the ribosome. So that DNA stays in the nucleus or in the nucleoid region if you're a prokaryote. And instead of taking a chunk of DNA, we're going to make a copy of it and send that copy. And that's to maintain the integrity of the DNA and make sure nothing happens to it. We also have ribosomal RNA, which along with proteins is going to make up the structure of the ribosome. So we'll get to that later in the lecture during the chapter 11 materials. Then we also have transfer RNA. Transfer RNA's job is to be the translator between the messenger RNA code and the amino acids. And we'll talk about how its structure helps it do that and what exactly it's doing. Again, that's in chapter 11. Please refer to table 10.2. If I don't hand it out in class, go check it out in your textbook. It has a description of what some of the other functional RNAs do. So they function in RNA processing, which is something specific to eukaryotes that happens between transcription and translation. It helps with regulation of transcription and translation. So essentially, it helps with gene regulation. And then there are a few other functional RNA jobs in a cell. One additional one that we're going to talk about later in just a couple weeks in lab are CRISPR RNAs. These are RNA molecules that help guide specific endonucleases to sequences within a genome to cut it up. It's basically part of a really cool bacterial immune system that can also be used for editing. So we'll talk about that a little bit later. Our goal here in this chapter is to discuss how we get from a genotype to a phenotype. And if you recall from our discussions of Mendelian genetics, we talked about a genotype as the set of alleles that an individual has, and that is a reflection of the particular gene that is encoded at a particular location on a chromosome. And that set of alleles produces the physical characteristic in the organism. So what we're really doing here is taking our information in our genes, whatever version that may be for whatever allele, and using it to produce a phenotype. We've talked about the definition of a gene a couple of times. When we were talking about Mendelian genetics, we were talking about how a gene produces a particular trait or characteristic of an organism. And then in chapter 8, we defined a gene as a nucleotide sequence, a specific set of nucleotides that is going to eventually produce a phenotype. So here we're going to get even more specific, and we're going to define a gene as a set of nucleotides that specifies the amino acid sequence of a protein. Now, I'm going to put a little asterisk here because it doesn't always specify the amino acid sequence of a protein. Sometimes instead it specifies a functional RNA molecule. So we won't always actually get a protein out of this. So actually a better way to say this is a gene is a set of nucleotides that specifies an amino acid sequence or a functional RNA sequence. But this is our textbook definition for right now, so that's what we're going to go with. Remember that a gene has to be replicated in order for it to be able to be passed to the next generation. And in that process of replication, there is potential for variation. And we'll see in chapter 13 that when we have variation in our DNA, this leads to variation in our phenotypes, which leads to variation in a population. And then lastly, a gene has to be expressed. So we're going to talk about how this process of gene expression actually takes place. And if we're going to build a protein, there are two steps to this process. The first step is transcription. And then the second step is translation. During the process of gene expression, we are going to start with the information in our DNA. So this is going to be our gene sequence. In other words, this is going to be our genotype. And then the end result is going to be a phenotype. Now that end result can be a protein, or it can also be a direct-to-function RNA molecule. And that direct-to-function RNA molecule is still going to somehow affect the phenotype of an individual. So the first step in our process, no matter which type of end product we're going to produce, is going to be the process of transcription. And in the process of transcription, we use the message encoded in our DNA to make a copy of DNA as RNA. And this specific RNA is messenger RNA. And this messenger RNA is going to take the message, the DNA message, to the ribosome, where it is going to be used to produce a polypeptide. Now, a polypeptide is an amino acid sequence, or let's say an amino acid chain. And this amino acid chain is put together during the process of translation. Now that amino acid that is initially produced is eventually going to be made into a protein. And that protein is going to then take on the specific function for the phenotype. Now there's a little step in here that we're kind of going to gloss over and kind of ignore, which is how we get from that amino acid chain or polypeptide to that functional protein. Basically what happens in the cell is that some folding takes place, the protein's modified based on functional groups that need to be tagged or removed, or slight modifications in order to produce its final function. So when we say our end result is a protein, we mean our end result is a protein, but the actual immediate first result of translation is going to be the amino acid chain. Our other option is to undergo transcription, taking our DNA and converting it into a functional RNA product. So functional RNAs have a variety of purposes in the cell. They are different from messenger RNA in that they are not used to make a protein. So we are not going to get that polypeptide. Instead, this RNA product, this functional RNA, is going to have a job in the cell. Now, sometimes it is gene regulation. Sometimes they're made into transfer RNAs or ribosomal RNAs. There are a variety of different things that these functional RNAs can do. But even though they're not being made into a protein, they are still generating a phenotype. This time, there's no second step. and they just are direct to function. We're going to start discussing our process of gene expression by going through the mechanism of transcription. And the goal of transcription is to produce a messenger RNA molecule from a DNA template. Now, there are a few things about this process that are similar to DNA replication, but there are a few things that make it very, very different. The first one is that RNA molecules are synthesized that are complementary and anti-parallel, just like in DNA replication. But we only copy one strand of the RNA molecule. That's because messenger RNA is single-stranded. And actually, even within the same region of DNA, you could code two different genes on opposite strands. So we could code a gene going this direction on one strand, this direction on another gene over here, and so on. So you can have overlapping genes on opposite strands because the sequences are complementary, which means they're not the same. So you can actually have sequences on both strands in the same region coding multiple genes. The messenger RNA is made from the template strand. We're going to talk about what that means in just a minute, but the strand that we use to make the complementary messenger RNA is going to be the template strand. And we'll talk about how you figure out which one is the template strand in just a minute. So here we've got just a quick example of a transcription fork. We've got the double helix opened up. You can see our sequences on either strand. And then you can see this green messenger RNA that comes in is being built in the 5' to 3' direction based on complementary base pairing. We're putting in that uracil because there's no T in RNA. And then since we're only using one strand, we get a single-stranded messenger RNA molecule. And one thing that is going to be sort of a theme with RNA molecules is the structure of the molecule implies the function. Or dictates, I think is a better word, dictates the function. So we have to have a single-stranded, straight, unfolded molecule of messenger RNA because that straight, flat messenger RNA has to fit into the ribosome. So it actually cannot take on a three-dimensional structure, otherwise it can't be translated. Let's take a look at our transcription unit. These are all the sequences that play an important role in either transcribing or transcribing and then translation. So as part of our transcription unit, we have a promoter region. Now the promoter region is a regulatory region that helps with control of gene expression. So we'll talk about this in a little bit more detail in chapter 12, coming up after this unit exam. But basically, whether or not a transcription factor or an RNA polymerase is able to bind to a promoter is going to determine if a gene turns on and when a gene turns on. So sometimes that regulation is due to binding of a regulatory protein, or sometimes it is due to the release of a regulatory protein so that the transcription factors and RNA polymerase can come in and bind. Now that promoter region is located upstream of our coding region. Our coding region is the region of DNA that is, RNA, yes, DNA, that is going to be used to make the messenger RNA that is going to be used to produce your protein or the functional molecule, depending on what type of gene we have. And then downstream or after the end of our specific gene, we have the termination site. So the termination site is going to signal the end of the messenger RNA, but also within that termination site, there's going to be a terminator. The terminator is going to be the stop region for translation. So within this transcription termination site, we've got the end for both the messenger RNA and then the protein that we're going to produce at the end. So the promoter does not become part of that messenger RNA transcript because that's the regulatory region. That promoter is going to be essentially the same no matter what type of organism it is. And the reason for this is that the transcription factor can bind anywhere there's a promoter that needs to be expressed, and we don't need a specific transcription factor or RNA polymerase for each gene. The end result of processing through this transcription unit is that we're going to get our messenger RNA transcript. And we'll talk about the mechanics for this in just a minute. Our first phase of transcription is the initiation phase. In the initiation phase, essentially everything comes together. All of our components come together. And just like DNA replication, in order to be able to build an RNA molecule, we need nucleotides. So we need ribonucleotides because this is RNA. And just like in DNA replication, we need a specific version of the nucleotide called a nucleoside triphosphate. Remember that those triphosphates have 3 phosphate groups. And the cleaving of the bonds between the first 2 phosphates is going to release energy that can be used to catalyze the bond between our 5' and 3' end. So the triphosphates are there for energy, just like in our ATP molecule, which is adenosine triphosphate, which is a modified nucleotide, but it's being used for energy. We will also see another energy molecule called GTP, and this is going to be essentially the same thing. It is going to be a guanosine triphosphate, and it can also be used for energy. But for our transcription, we need ribonucleoside triphosphates. These are going to be adenine, uracil, guanine, and cytosine ribonucleotides. And we also need the bacterial polymerase, RNA polymerase. This is another hollow enzyme, which is a very big enzyme comprised of multiple subunits. Just like DNA polymerase was a hollow enzyme, so is RNA polymerase. And then we need a sigma factor or a transcription factor. A transcription factor is going to help the RNA polymerase bind to the promoter. And so that sigma factor is going to be super important in making sure that RNA polymerase is there, it's essentially installed correctly, and can start at the right location. I just noticed a typo. Okay, our bacterial promoter, where initiation is going to take place, is what we call a consensus sequence. A consensus sequence is a sequence that we find to be the same across many genes or across many organisms. And if not the same, very, very similar. So for example, there are some consensus sequences that are going to be the same in every single mammal or every single eukaryote or every single bacterium. And the reason for that is to reduce the need for a variety of different transcription factors, RNA polymerase, DNA polymerase, helicase, all of those things. They are sequences that we can use so that multiple enzymes can recognize where they are supposed to bind. So within the promoter, there are 2 consensus sequences, one that is located at 10 base pairs upstream from the transcription start site. So the transcription start site is going to be right at the edge of the promoter. We draw our gene sequence, our gene diagram. Again, we've got our promoter, and we've got our coding region, and we've got our termination site. So right here at the edge of the promoter and the coding region is going to be our start site. So this is where transcription is going to start. Now, 10 base pairs upstream, or at -10, if we call this the 0, 10 base pairs upstream, we're going to find our first consensus sequence. This is called the PRIBNOW box, and the PRIBNOW box mostly consists of T's and A's. The reason for this is, remember that T binds to A with only 2 hydrogen bonds, which means this is going to be easy to open up the helix, compared to if we had a bunch of G's and C's where we've got 3 bonds holding them together. And so that is going to actually be the location where RNA polymerase is going to open up our double helix so that it can start transcription. And then up a little bit further at the -35 sequence, we have another consensus sequence that's really going to help our sigma factor bind, which then means our RNA polymerase can bind. So we need both of these consensus sequences to make sure that the sigma factor gets there, the RNA polymerase gets there, and then the RNA polymerase can open up at the right site so that it can start at the right nucleotide. So here we have our promoter region with our 2 consensus sequences, one at -35 and one is -10 base pairs upstream from our transcription start site. So the sigma factor is going to act as a guide for RNA polymerase. So that sigma factor protein is going to bind at that consensus sequence, and then it's going to guide in RNA polymerase. And then RNA polymerase is going to open up our double-stranded molecule here at the PRIBNOW box. And then once it is open, it can start transcription at that start site. So none of this promoter region is going to be transcribed, but everything from that start site forward until we get to the terminator is going to be transcribed into messenger RNA. There is no primer required for RNA synthesis. Remember, for DNA synthesis, DNA primase came in, laid down a little RNA primer to start replication. RNA primase could do that because RNA primase is an RNA polymerase, and it can add in the complementary nucleotide without a 3' end to attach it to. And then once we have the very first base, then RNA polymerase can continue adding on to produce that messenger RNA. Same thing with the DNA primase from DNA replication. Once we have that first base pair, then we get a short strand of RNA long enough for the DNA to bind and then start adding on DNA. But we are talking about transcription, so we have our sigma factor, which is basically a smaller protein that's going to guide that RNA polymerase into the right promoter region, get it all set up and ready to start transcription. Once all that is done, then we're ready to begin producing our messenger RNA and elongating it. So now we're ready to start building our messenger RNA. We have our RNA polymerase bound at our start site, and it's going to begin adding nucleotides in a 5' to 3' direction. And it's going to continue to do that. As it does, RNA polymerase is going to push open this replication fork and expand that replication fork. Behind it, as the messenger RNA is produced, the strand of DNA actually comes back together and it binds back, forms its double helix. When it does that, it pushes the messenger RNA off of the DNA strand. So the messenger RNA is basically making a trail off of the fork that the RNA polymerase is creating. And as RNA polymerase moves along, it's going to keep pushing open this fork here and extending our messenger RNA. In class or on Brightspace, you should have a handout that shows this process in a little bit more detail, or you can check your textbook because your textbook has detailed images of each of these steps. This one is a little bit simpler one, so you can kind of see what's happening. So finally, we reached the end of our RNA coding region. And in bacteria, there are two different ways that transcription is terminated. The first is called row-dependent termination. Row-dependent termination requires something called a row factor. Now, the way that the row factor works is that it binds to a spot in the messenger RNA called the rut site. And it binds at that rut site and then basically moves along the messenger RNA until it reaches the transcription bubble with the RNA polymerase attached to the messenger RNA. As it continues on, it begins to unwind the DNA RNA pairing from one another. And so once that row factor gets up to the region where RNA polymerase is currently working, basically it catches up, it starts to detach the DNA and the RNA. And so it causes the messenger RNA to come off, and then the RNA polymerase releases as well. The second type of termination is called row-independent termination. Now, this one is not based on any factor that binds to the messenger RNA. Instead, it depends on the structure of the messenger RNA itself. And the way that this one works is that a region is transcribed onto the messenger RNA that has complementary repeats. And so what happens is that it causes a hairpin loop in our messenger RNA, which basically makes the DNA RNA complex unstable. So that causes the messenger RNA to release from the DNA comes back together, and the RNA polymerase releases as well. So finally, we get to the end. Our messenger RNA is ready to be translated. And so if this were a bacteria, translation could begin as soon as that messenger RNA begins to be produced. So as soon as the mRNA begins to pull away from the DNA, then a ribosome can bind and start translating it. In a eukaryotic cell, which is what we're going to continue with, there's a few modifications that need to be made before that messenger RNA can go to the ribosome. And so our next video is going to focus on how this messenger RNA that we have made, which is actually a pre-mRNA, is processed into a mature mRNA that is ready to go to the ribosome.